Look at this pill submerged in acidic fluid. It isn't dissolving. Modern active pharmaceutical ingredients, or APIs, are specifically engineered to be chemically stable. They must survive the harsh, highly acidic environment of the human digestive system to reach their target organs intact. But that exact stability creates a secondary problem. After the patient excretes the drug, these resilient molecules travel through local sewage systems. Because they are designed not to break down, they easily survive standard wastewater treatment processes and flow directly into rivers and streams. For decades, the pharmaceutical industry focused almost entirely on pharmacovigilance, monitoring a drug to ensure it is safe for the human taking it. Now, the scientific community is being forced to practice eco-pharmacovigilance, tracking the risks those same drugs pose to the broader ecosystem. The biological targets these drugs act upon, like specific hormone receptors or enzymes, are evolutionarily conserved. That means the exact same receptors exist in other vertebrates. A drug meant to manage human anxiety or regulate human hormones will inadvertently trigger the same physiological responses in wild fish populations. We are moving toward a safety model that monitors a drug through its entire environmental life cycle. It requires us to account for a medicine's impact on the ecosystem with the same scientific rigor we apply to the patient. To enforce that life cycle safety, pharmaceutical companies must submit an environmental risk assessment, or ERA, before a new medicine can be sold. Two primary gatekeepers dictate how this is done: the European Medicines Agency and the U.S. Food and Drug Administration. This is the European Medicines Agency's 2024 ERA guideline. Notice the effective date of September 1, 2024. Under this framework, a complete environmental dossier is mandatory for every single human medicinal product applying for market authorization, whether it is a brand new drug or a generic. Across the Atlantic, the U.S. Food and Drug Administration operates under a different legal framework called the National Environmental Policy Act. Here, the FDA relies heavily on categorical exclusions, meaning most new drugs are exempt from submitting an environmental assessment if their expected concentration in the water is deemed low enough. The EMA triggers environmental testing if a drug's surface water concentration hits 0.01 micrograms per liter. The FDA's threshold is 100 times higher. But antibiotics and endocrine disruptors bypass these limits entirely. Because of their potency, both agencies demand testing regardless of concentration. By setting its baseline trigger so much lower, the European system captures a wider net of pharmaceutical compounds, prioritizing the long-term, chronic risks that trace-level chemical exposure poses to aquatic life. Here in Section 4.1 of the EMA guideline, we find the starting point for calculating that risk, Phase 1. This is a screening phase designed to find the predicted environmental concentration, or PEC, of a drug in local surface water. This equation calculates the PEC assuming a worst-case scenario. It takes the total amount of the drug consumed in a region, multiplies it by market penetration, and divides that by the number of inhabitants and the standard wastewater dilution. If that final PEC number exceeds the 0.01 threshold, the drug moves to Phase 2. This requires physical laboratory testing on 3 distinct levels of the aquatic food chain: algae, daphnia, and fish. The goal is to find the exact concentration where the drug produces no observable adverse effects on these organisms. But laboratory conditions do not perfectly mimic real rivers. To account for that uncertainty, regulators apply assessment factors. As this table shows, if a company only has acute, short-term data, the lab result is divided by a massive factor of 1,000. If they provide high-quality, long-term, chronic data across all 3 species, the factor drops to 10. The final step is determining the risk quotient. This ratio pits the predicted environmental concentration against the safe no-effect concentration. If the predicted exposure in the environment outweighs the safe limit, the risk quotient pushes above 1.0. A score above 1.0 confirms an ecological hazard. This specific mathematical framework removes ambiguity, translating abstract environmental concerns into a strict enforceable limit that the pharmaceutical industry must operate within. Watch how this molecular model responds to light and water. When a drug hits an unacceptable risk quotient, manufacturers are pushed towards green chemistry, designing active ingredients that deliver their medical benefit in the human body, but rapidly fragment into harmless organic compounds once exposed to the elements. The responsibility extends downstream. Mitigating high-risk medicines require robust community take-back programs to prevent people from flushing unused pills, alongside expensive municipal upgrades to advanced wastewater treatment plants capable of capturing stable APIs. To accelerate this process, regulators are pushing for global data transparency. As outlined here, the EMA supports a centralized ERA database. By sharing environmental data across the industry, companies can accurately assess ecological impacts without unnecessarily repeating toxicity tests on live animals. The regulatory consequences are intensifying. Under proposed reforms to European pharmaceutical legislation, failing to present a complete environmental risk assessment or refusing to implement mitigation measures could result in the outright denial of a drug's market authorization. Ecopharmacovigilance ensures that the pipeline of modern healthcare does not end at the patient. It links the physiological safety of the individual directly to the biological integrity of the planet's water systems. Modern healthcare relies on these chemical innovations. The next challenge is ensuring the science used to cure us is compatible with the world that sustains us.